In a world where the human genome is not sequenced and A, T, C, and G are still unknown, the Human Genome Project will come along to solve the conundrum. December 20th, 2006, Irene D'Alessandro, Angela Chan, Dion Dawkins, Gavin Dane Ryan, Shamim Hussain, and Kalyani Dar will attempt to simulate their groundbreaking and historic work. It all began with the Sanger sequencing method. This method involved taking 2 strands of DNA and after taking them, isolating specific bases and cutting out the DNA at those bases. This was done for each of the 4 bases. This sequence was very, very long and arduous. Thus, the private Human Genome Project was launched. This involved blasting the sequences, such as this stack of paper, which represents our human genome, blasting it into several different sequences. Then, overlapping sequences would be put together to form the human genome. However, redundancy was found in these genomes, which made this process ineffective. However, the gaps were filled with public DNA. This process was similar in that it involved breaking up the DNA and blasting it apart, just like in the private Human Genome Project. However, it was different in that it used marker sequences, represented by these red tabs. Marker sequences allowed us to put the human genome into specific sequences and to know what order they would go in. For example, the DNA here with these marker sequences, let's say, means the first sequence. Matching up with this sequence is this. You have a whole section of DNA. Yet another sequence. This one. Put in right here. These sequences as well. However, there were discrepancies, still gaps in the process. These gaps were being worked upon to fill up and complete the human genome. December 20th, in movie theaters near you, the Human Genome Project.